Hello, I'm Who Are We, and on today's episode of The Cutting Edge, we will be investigating a study titled Seeing Beyond Violet, UV Cones Guide High-Resolution Prey Capture Behavior in Fish. The study interrogates one of the most anthropocentric assumptions in visual neuroscience, the notion that the limits of human perception demarcate the meaningful boundaries of the visual world. So this study was done by Johan Westo and Petri Alla-Lorlila. It was obviously done in Finland at Aalto University. So, let's get into it. The focus will be on a recent and elegant body of work examining the Nino Riro, or zebrafish, and how their ultraviolet vision, mediated by a discrete population of UV-sensitive cones, orchestrates a sophisticated behavioral sequence, high-resolution prey capture. We will explore 3 levels of analysis: cellular and receptor level specialization, retinotopic functional asymmetries, sensio-motor integration, and ecological relevance. 1. The architecture of seeing specialized UV-sensitive cones. Most vertebrate visual systems operate within a spectral window bounded by approximately 400 to 700 nanometers. But zebrafish, and indeed many aquatic species, possess UV-sensitive cones peaking around 360 nanometers. These are not mere spectral extensions of color space. They are functional adaptations, evolutionarily tailored to the optical properties of their environment. The key point: in larval zebrafish, UV cones are concentrated in a strike zone of the retina, analogous to the fovea, maximizing spatial acuity where it matters most, the upper frontal visual field. Physiological adaptation. These UV cones exhibit faster kinetics and higher photon capture efficiency, a necessity in dim underwater UV environments. This raises the broader question: to what extent is color merely a convenient category imposed by the human visual system, rather than an intrinsic property of the world? We have functional asymmetries. Retina as a computational grid. The spatial non-uniformity of zebrafish UV vision maps closely onto ecologically relevant behavior. The strike zone, rich in UV cones, is biased towards detecting prey objects, typically planktonic organisms that reflect UV light strongly. Evidence from 2-photon calcium imaging. Neural recordings show enhanced activity in UV cone pathways when prey-like stimuli, small UV bright dots, are presented within the strike zone. Retinotopic mapping. The spatial specialization is not simply an anatomical curiosity. It is embedded in the very structure of the retinal ganglion cell output. The visual field is functionally warped to amplify ecological salient input. This asymmetric processing calls to mind earlier discussions in visual neuroscience regarding the primate visual cortex and the cortical magnification of the fovea. However, we observe a retinal level magnification of ultraviolet contrast in the region of behavioral relevance. UV-guided prey capture, sensory input as behavioral predicate. What does it mean to see in the context of predation? In larval zebrafish, visual processing is not passive reception, but an anticipatory computation leading to an action plan. Behavioral assays reveal strike initiation is significantly impaired when UV stimuli are filtered out. Accuracy and speed of strikes correlate with the density and activation of UV cones in the strike zone. Behavioral specificity. The zebrafish do not generalize UV-driven behavior to other photoreceptor classes. This is not a redundant channel, it is a dedicated one. This is not merely an example of sensory-guided behavior. It is a real-time instantation of high-resolution sensio-motor coupling behavior mediated by a photoreceptor class that humans do not even possess. In philosophical terms, we might call this Umwelt. Jacob von Esox calls concept the idea that each organism constructs a subjective perceptual world. UV cones in fish exemplify how different biological substrates yield radically different perceptual ontologies. Theoretical implications. Perception as ecologically tuned inference. We must resist the temptation to interpret vision as a generic computational pipeline. What this research makes clear is that sensory systems are sculpted by ecological niches. The zebrafish visual system does not merely reflect its world, it predicts it, by weighing high-fidelity input in behaviorally critical regions. This invites a reconceptualization of visual perception as an inferential process shaped by evolutionary priors. In this framework, photoreceptors are not merely light detectors, they are statistical priors encoded in tissue. Behavioral strategies like prey capture are the model outputs of an organism's predictive coding hierarchy. The conclusion. A UV mirrors for neuroscience. The UV cones of a zebrafish are more than a curiosity. They are a challenge to the anthropocentric assumptions in vision science, and a reminder that perception is not an absolute, it is a relational, contingent, and ultimately goal-driven. If we want to build a truly general theory of perception, we must look beyond the visible, both literally and conceptually. Now, of course, I also want to know, is this specifically exclusive to the zebrafish, or can we study this in other fish? And I will hopefully be finding that out very, very soon. But anyways, thank you for listening. I'm Uwe, and this was another episode of The Cutting Edge. And if you want to learn more about fishing, please take a look into this series that I've linked in the description. It's a book series called A Systematic Approach to Fishing. The most recent book is A Systematic Approach to Northern Pike Fishing, a data-driven guide to pike ecology, equipment engineering, and tactical fishing strategy. Again, the link is in the description, and thank you all for watching this video.